What if your next vaccine wasn't delivered with a needle, but was instead printed, almost like a 3D object? Imagine a small, painless patch embedded with microscopic needles, created on demand in a matter of hours. This isn't science fiction. It's a glimpse into a revolution happening right now in biochemical engineering. A revolution that promises to make life-saving medicines and vaccines incredibly fast, remarkably cheap, and astonishingly precise. To understand why this is so game-changing, we first need to look at the challenges we face today. The problem is that traditional biochemical production is a monumental undertaking. It's slow. Developing a new biologic, like an antibody or a vaccine, can take over a decade. It's a marathon of research, development, and rigorous testing, with countless hurdles along the way. It's also incredibly expensive. The process requires enormous, specialized factories filled with giant steel vats. Scaling up from a small lab discovery to mass production is a billion-dollar gamble. Think of it like trying to scale a kitchen recipe for a single cake to bake a million cakes at once. It's not just about using more ingredients. The entire process, the equipment, the physics, it all changes, and failure is costly. And finally, the products themselves are often fragile. Many advanced medicines are delicate proteins that must be kept in a constant, refrigerated state from the factory to the patient. This is the cold chain. A single break in this chain, a power outage or a logistical delay, can render a life-saving shipment completely useless. This fragility limits access, especially in remote or low-resource areas. But what if we could bypass these bottlenecks entirely? The breakthrough comes from a new suite of biochemical engineering tools that are fundamentally changing how we make medicine. First, there are microfluidic reactors. Imagine shrinking a massive chemical plant down to the size of a credit card. These labs on a chip use tiny channels to mix and react chemicals with incredible precision. This continuous flow process is faster, uses vastly fewer materials, and gives scientists unprecedented control, allowing for rapid iteration and optimization. Next, we have AI-guided design. Instead of relying on trial and error, scientists can now use artificial intelligence to design and predict the behavior of new proteins and enzymes. AI algorithms can sift through billions of potential molecular structures in seconds, identifying the most promising candidates for a new drug or vaccine, dramatically accelerating the discovery phase. Then there's cell-free synthesis. Traditionally, we use living cells like bacteria or yeast as tiny factories to produce biologics. But this can be slow and complex. Cell-free systems take only the essential machinery, the DNA instructions and the molecules needed to read them, out of the cell and put them into a test tube. This creates a clean, controllable, on-demand production system that can be freeze-dried for stability, eliminating the need for a cold chain. Tying it all together are modular bioprinters and lab automation. These robotic systems execute the complex workflows with tireless precision, running experiments 24/7. They are the hands that operate these new tools, collapsing timelines and costs by creating a seamless automated pipeline from digital design to physical product. So, how does this all work in practice? It's a powerful cycle. Design, build, test, and learn. A scientist uses AI to design a new molecule. That digital design is sent to an automated system that uses cell-free synthesis inside a microfluidic reactor to build it. Integrated sensors then test the product in real time. The data feeds back into the AI, which learns and refines the design for the next cycle. This entire loop can happen in hours, not months. The real-world applications are staggering. During a new viral outbreak, scientists could sequence the virus, design a corresponding mRNA vaccine using AI, and produce the first test batches in a portable, on-demand biomanufacturing unit within a single day. These microfactories could produce insulin precursors in a local pharmacy or create point-of-care diagnostic sensors for a doctor's office, tailored to a specific patient's needs. Of course, with great power comes great responsibility. Safety and quality are paramount. These new systems are being designed from the ground up to meet and exceed the stringent standards of good manufacturing practice, or GMP. Every step is monitored, every batch is tested, and every piece of data is logged, ensuring complete traceability and integrity from the first line of code to the final dose. The ultimate payoff is democratized access to healthcare. By decentralizing manufacturing, we can bring production closer to the people who need it most. Imagine a world where a remote clinic in a developing nation can print its own vaccines and medicines, tailored to local needs without relying on a fragile, expensive global supply chain. This means faster response to outbreaks, lower costs for everyone, and countless lives saved. This transformation is also creating a new generation of careers. The biochemist of the future is also a data scientist, a programmer, and a robotics engineer. This convergence of biology, data, and automation requires a new set of skills, opening up exciting pathways for students and professionals willing to learn and adapt to this new paradigm. Looking ahead, we are entering an era of programmable biology. Just as we program computers with code, we will soon program biological systems to produce nearly any molecule we can imagine. A distributed network of these smart, agile biofactories could form a global immune system, ready to respond to any threat, anywhere. The lab is getting smaller, smarter, and seriously game-changing. The future of medicine is not in massive factories, but in small, intelligent devices. It's a future that is more accessible, more resilient, and more personalized than ever before. Ready for the biotech revolution?